Hi, my name is Tilanka, and I'm going to talk to you about the pipeline I created to be used by evolutionary biologists who are interested in understanding the protein domain evolution under different contexts. You will find proteins in any biological system, for example, plants, animal, bacteria, even in fungi. If you zoom into these proteins, you will see there are some structural units inside proteins. For example, in this protein, you will have three structural units. But later, scientists found out that these small structural units also carry out separate functions, and they also tend to evolve separately from the main protein. And they named these structural units as domains. If you think about a human, there are around 400,000 different proteins. And if you think about a tomato plant, there are about 50,000 different proteins. And in bacteria, there are about 2,500 separate protein present. But if you look at the above example, there are more than one domain per protein. So if you think about the domain number per species, it's really a head scratcher, isn't it? In my research project, I'm interested in understanding the protein domain evolution as well as to make inferences about the impacts of domain evolution on several different phenotypes. The general guideline to infer domain evolution is first, you need to identify what types of protein domains present in a particular species. Even if you have this information, you will still have a heterogeneous sample. So you need to separate these domains based on sequence similarity. Domains which are more similar in terms of the amino acid sequence are known as domain families. So the next step is to separate out domains into domain families. Finally, when you have this information, then you can make phylogenetic trees for each domain family. And based on phylogenetic tree, you can make different inferences. In this pipeline, I automated the process of separating out different domains families, also optimized the automation of phylogenetic reconstruction process. However, to do the step one, which is this one, we need to run program called Hammer, which I will talk to you about more later. Let's look at what each script is doing. The first one is domain finder. If you remember, I told you before to run this script, you will need an output from a software called Hammer. In this Hammer software, there's a module called Hammer Scan that scans your sequence against a profile database, and it will give you an annotation which is simply a table describing things such as where the domain is, places they are found in the protein, and the alignment details. So in the perfect scenario, where you will have a single type of domain per protein and where the HMM model would align with the domain part of the protein like this. But in real life, domain tend to duplicate within the protein where you will have complex cases like this. So we need to figure out if all these blue block boxes are really duplicated domains or they are misannotations, depending on the annotation given by the HMM scan. So depending on a threshold value, this script find out the duplicated domains from the misannotation and single domains. You can read more on how to set the threshold and the details on the threshold in the documentation part in GitHub. Once you do this, then you will categorize domain families, and then using domain families, you can make phylogenetic trees. The next script will produce and submit a job submission script into HPCC, which will make phylogenetic trees using the software called RAXML per domain family. We know when running a bootstrap maximum likelihood analysis, we at least iterate the analysis for 1,000 replicates. So the idea behind running a single job script is using this script to estimate the time that is taken to run a tree. Because we all know each domain family would take different time to run the phylogenetic tree, depending on how big sequence is and what is the number of sequences per domain family. Then By implementing the make final tree.py script, it can estimate how many jobs can be run within 2 hour time scale, which is the sweet spot for HPCC. Then using a job submission array to submit optimized number of jobs where we have 1,000 replicates in the end. So that's what this script is doing. Hope you enjoyed this video and hope you will use this domain finder software more often. Thank you.